So, of course, most of you are very well familiar with the Ediacara biota, but just a very brief overview. The Ediacara biota is our earliest fossil evidence for a complex, macroscopic, multicellular community. The Ediacara biota occurs in deposits worldwide in strata that were deposited in the wake of the snowball Earth cryogenic glaciations, which represent our most severe perturbation of the climate system in Earth history. Ediacara organisms themselves occupy a wide range of sizes, from the millimeter to the meter scale. They were almost entirely soft-bodied. In terms of their phylogenetic affinity, they've proved somewhat elusive. They don't easily fit into any of our modern classification schemes, but they were undoubtedly complex, both morphologically and ecologically. And they include a wide array of diverse body fossils, and also trace fossils, such as helminthoid ignites. And the presence of these trace fossils tell us that regardless of the affinity of the more enigmatic body fossils, animals were certainly present in these ecosystems, because in order to excavate sediment and form a burrow, you need to have a coelom. You need to be a bilaterian animal. And one of the other features that makes the Ediacara biota so distinctive is the presence of widespread, diverse, and complex mat ground structures, or what we term textured organic surfaces, which are macroscopic, patterned, or iterated organosedimentary textures. And as you can see, these textured organic surfaces really include a considerable morphological diversity, far greater than we typically think of when we think of myths or wrinkle marks or kinea. And in fact, many of them are complex enough that we can be reasonably certain that these so-called mats included not only prokaryotic communities, but also likely eukaryotes. So the complexity and diversity of these textured organic surfaces really sets the Ediacaran apart in Earth history. And another reason that these textured organic surfaces are significant are because, as popularized by Dolf Zeilacher and others, they record interactions between the Ediacara organisms and their substrate. And particularly, they reveal that many of these Ediacara organisms had specific mat ground adapted ecologies. They had life modes and feeding strategies particularly well suited to life on an organically bound substrate. So, for instance, we have organisms like Kimberella, which is associated with these linear radiating structures, or radialicness, from which we interpret it to be some sort of a possible stem group mollusk, which made its living by scratching or radula scraping of the mat ground surface. Or, for instance, we have mat stickers, organisms such as Claudina, which likely stabilize themselves within an organically bound substrate. Other Ediacara organisms appear to have encrusted the microbial mat, and still others made their living through mining of the organic matter associated with these mats. And in addition to mediating the ecology of these Ediacara organisms, these textured organic surfaces are significant because they're intimately associated with the taphonomy of these organisms. So the Ediacara biota is no doubt, as well accepted by all of you, very much a taphonomic phenomenon. The fact that we find these soft-bodied deposits worldwide suggests that some sort of anaxialistic taphonomic conditions were at work in these Turbo-Neoproterozoic settings. And particularly striking, many of these Ediacara fossil deposits are characterized by a highly unusual mode of preservation, and that is casts and molds preserved on the basis of sandstone beds, as you can see, particularly well preserved in the Ediacara member of South Australia. And in the Ediacara member, we interpret this mode of preservation to reflect a unique combination of circumstances, of sedimentological processes, of the presence of these textured organic surfaces of varying types, and also different tissue integument of these benthic organisms. So, for instance, in the Ediacara member, if you envision you have your benthic community living on or within these organically bound substrates, which gets suddenly buried, those organisms that are composed of more resistant tissue structure are less likely to collapse, and therefore the sand bearing them drapes around them, resulting in the formation of a negative relief external mold on the basis of the overlying bed. So for instance, Dickinsonia is always characterized in the Ediacara member by negative relief external mold preservation, and from that we interpret it to have been composed of a more resistant integument. Conversely, other Ediacara organisms are interpreted to have been composed of less resistant tissue structure, and these organisms would have been more susceptible to rapid decay and collapse, and the resulting hollow would have been filled in by the sand, resulting in the formation of a low positive relief structure on the base of the overlying bed. So for instance, many of our tubular faunas, such as phylozoan, are characteristically preserved as positive relief structures on these sandstone bed bases. So it's really this unique combination of sedimentological, of organic substrate, and tissue integument factors that has shaped this anomalous taphonomic mode that we see in the Ediacara member. And it's important that it's really vital that we start to grapple in more detail with these long-standing taphonomic questions in order to not only get a better handle on what are the circumstances responsible for this particular exceptional preservation, but in order to better translate, in order to better reconstruct the paleobiology and paleoecology of these organisms. So we've had, for many years, the tendency to liken these assemblages to geologic snapshots or still lifes, and that is basically the idea that what we see in the fossil record is a direct reflection of life as it was on the Ediacaran seafloor. However, there's some degree of bias inherent in this perspective, in that traditionally we have tended to focus on the best preserved specimens, that is, the organisms that in life were composed of the most resistant tissue structure. So those organisms that are less well preserved, that is, in life they likely possess less resistant tissue, or those organisms that are less easily characterized, such as our textured organic surfaces, have tended to be overlooked. So in the end, we may find that taphonomy has played a very important role in filtering our view of the diversity of these ecosystems, and we need to have a stronger grasp of the taphonomic processes at work in order to understand whether taphonomy is artificially amplifying or diluting our picture of Ediacara diversity. And perhaps one of the best ways in which we can accomplish this is through holistic bed-scale paleoecological analysis. And we're fortunate in South Australia, and especially at Nopina Station, in that we have exceptional preservation and also exceptional exposure, which have allowed us to excavate beds and reconstruct them. And so to date, we've reconstructed over 300 square meters of Ediacaran seafloor, which allows us to, on a very broad spatial scale, but also a very detailed stratigraphic scale, compile all sorts of data on the body fossils, the trace fossils, the textured organic surfaces, the sedimentology, the taphonomy, and better understand the interactions between these various factors. And perhaps one of the most compelling examples of the manner in which we can combine data on body fossil morphology and taphonomy and on textured organic surfaces and understand how one mediates the other is the case of Aspidella. So Aspidella is a form genus for the holdfast of a frondose organism, a Charniodiscus type organism. And Aspidella is one of the most common fossils of the Ediacara biota. There are over 1,000 specimens at Nopina Station alone. And Aspidella is also one of the most variable of the Ediacara taxa. We find it preserved in a variety of modes. We see it as internal modes, as external molds, as casts. We see it preserved with a variety of morphotypes, a variety of morphological features. But one of the systematic relationships that we can observe due to this bed scale holistic analysis is its association with textured organic surfaces. And so we observe, in fact, that wherever Aspidella is associated with well-developed textured organic surfaces, well-developed mats of this tubular organism known as Phoenicia, so you can see sort of a matrix of Phoenicia here. Wherever Aspidella is associated with well-developed Phoenicia mats, it is preserved as an internal mold, capturing the base of the holdfast. In contrast, where Aspidella occurs in the absence of a well-developed Phoenicia mat with only a thin biofilm, what we instead get is wholesale removal of the holdfast from the substrate, yanking out of the frond, resulting in the formation of this biological tool mark that we term mop for its resemblance to the household cleaning aid. Or what we instead see is burial of the upper surface of the holdfast, and often entrainment, three-dimensional infill and entrainment of associated stalk fragments. So asphodel is really a compelling example of how association, how presence or absence of a particular textured organic surface can really dramatically shape taphonomy or morphology of just an individual body fossil. Ediacara fossils are also characterized by heterogeneous facies associations, and the Ediacara member has really been instrumental in allowing us to recognize that members of all three of the traditional Ediacara bioassemblages, the Avalon, the White Sea, and the Nama assemblages, which traditionally have been considered to be disparate either biogeographically or biostratigraphically, can in fact occur together in a single locality. They just tend to occur in different facies. So particular taxa are characterized by very particular facies associations. Interestingly, the textured organic surfaces are also characterized by very particular facies associations. Both the body fossils and the textured organic surfaces occur in a wide range of shallow marine environments, but there is considerable heterogeneity in where they are between fair weather and all the way down to below storm wave bays. So a particularly interesting example of a facies-specific textured organic surface is this structure we call micropucker. And micropucker is characterized by dense clustering of dimples and of interconnected ridges that form these discrete patches that are very distinct from the surrounding surface. And micropucker occurs exclusively on the basis of silty, laminated, and tool-marked sandstone beds associated with our sheet flow sandstone facies. So we interpret these beds to have been deposited below storm wave base, and it does not occur in any of the other facies. One of the other interesting features about micropucker is we commonly observe it in association with degraded or taphonomically deteriorated body fossils, such as around the margins and creeping over the edges of this folded, desiccated, and frayed Dickinsonia, or over the top of the scleritic elements of the spicular elements of our Ediacara sponge taxa, Chronocolina. So based on this observation that micropucker is superimposed upon these degraded body fossils, we interpret it to have been a post-mortem growth. So we're not looking at a structure that forms synchronously with the colonization of this surface by the body fossils. It was a post-mortem overgrowth. In contrast, we have the textured organic surface weave. And weave is characterized by these linear subparallel elements, which are connected at acute angles by transverse elements, forming these sort of Y-shaped or bird foot-shaped junctions and branching patterns. So you can sort of see where we get the name weave. It looks vaguely like a woven fabric. And weave, like micropucker, has a very strong facies association, but unlike micropucker, it occurs only on the basis of sandy, amalgamated event beds that are capped by oscillation ripples. So a much more energetic environment, somewhere between fair weather and storm wave bays. And like micropucker, weave is associated with body fossils. However, unlike micropucker, we find body fossils and very crisply preserved body fossils superimposed on top of weave. So for instance, here we have a very nicely preserved Sprigina superimposed upon this patch of weave. And here on the left, we have Dickinsonia footprints, which you can basically think of as long-term resting traces for Dickinsonia superimposed on top of a larger patch of weave. So this tells us that unlike in the case of micropucker, the formation of weave preceded at least some of the stages of colonization of this particular bed and was not a post-mortem feature, but actually was some sort of a background synchronous with colonization by these macroorganisms. And when we consider weave on a bedding scale, what we see is that it is actually quite incredibly abundant, and the majority of this fossiliferous surface is covered by weave. So this is one of our most fossiliferous beds. It's about 9 meters by 5 meters. Each of these yellow circles is the centroid of an individual patch of weave. And if you're wondering why there are patches of weave where there's no bed, these photos were taken prior to later excavation and augmentation of this bed with considerably more fossiliferous material. But suffice it to say that you can see very broad spatial distribution of weave across pretty much the entirety of this bed. And when we measure the orientation of these individual patches of weave, we see that there actually is a fairly strong alignment. And in fact, this alignment corresponds very closely with that of other structures on the same bedding plane, such as mop, which remember is our biological tool mark, that results when we remove a holdfast from the substrate, and also these other features that we term lifts, these indentations along the margins of Dickinsonia, which are especially characteristic of Dickinsonia on this bedding plane. So we see not only is there a tight alignment within weave and within each of these structures, but all three are roughly characterized by the same alignment. So this would suggest a similar current-mediated origin for all of these. Mop we know to have been current-derived. These indentations in Dickinsonia have been interpreted by Scott Evans and colleagues to represent current-mediated deformation of the margin of Dickinsonia, and this would suggest that weave, too, has some sort of a current-mediated origin. And in fact, we interpret the patches we see of weave to represent areas of the mat which were partially picked up, partially disturbed and lifted up by these currents, not entirely severed from the substrate, but intercalated with sand, resulting in crisper preservation of these particular portions of the mat against a ground mass of more muted biomat.
2 minutes.
So in conclusion, what can we say about the Ediacaran substrate? Well, based on the very strong diversity, the abundance, and the complexity of these textured organic surfaces, I think we can safely say that the Ediacara substrate was extremely heterogeneous, and that these textured organic surfaces played a very strong role in both the ecology and the taphonomy of these surfaces. Not only did these textured organic surfaces play a role in the stabilization of this substrate, providing a more hospitable place for colonization in a fairly energetic environment, they also provided the conditions under which unique life modes, background-adapted feeding and life ecologies could develop, such as under mat mining or mat scratching. And they were also very much associated with the biostratinommy, with the taphonomy of these Ediacara organisms, both at the scale of individual structures and at the scale of entire bedding planes and entire facies. And I think perhaps just a final concluding thought that these textured organic surfaces really play an instrumental role, or will continue to play an instrumental role in our ability to reconstruct the evolution of the benthos from the terminal Neoproterozoic through to the early Phanerozoic, and that they may actually provide us with the tool to answer some of our long-standing questions, such as why did the Ediacara biota disappear at the end of the Ediacaran, and what were the conditions that especially optimized the radiation of complex animal life in the early Paleozoic. I'd like to thank various sources of funding and people, and you for your attention.